thanks for being here. I'm making the presentation on behalf of Alessandra Basso. She had a little emergency to get to. So be patient with me. It's my second day at Purolite. So, we're going to discuss who Purolite is and the type of product that they are offering to the life science industry. So Purolite is actually the second largest ion exchange resin manufacturer in the world. They're very focused on ion exchange and purification, from chromatography to large-scale purification. A lot of people know them in the purification of water or chemical, but they also have a department in the life sciences that's more focused on the pharmaceutical part of their business. So this explains some of the Purolite Life Science product line they have. So they have the Presto, agarose. This is mainly for large molecular weight protein separation and antibody purification, such as the protein A, affinity chromatography, cation, anion exchange resins, and so on. And it's based again on agarose. We also have the biocatalysis part of the product line. Biocatalysis is where we immobilize enzymes on polymethacrylate or polystyrenic-based matrices to achieve, enzymatic reactions repeatedly and reuse the same enzyme for hundreds to thousands of cycles. We also have the resin that are used also for peptide oligonucleotide production. When you're synthesizing your peptide or oligo, they would use a resin with HMAC and so on it to try to build up these peptides and oligos. But what I'm presenting here mainly is on our fourth product line, the Chromalite synthetic resins. So what are the Chromalite synthetic resins? They're mainly polystyrene or divinyl benzene resins, most of the products. Some of them are non-functionalized, while others are ion exchange. They're typically used for the small molecule separations. The Chromalite's CGC, for example, is the cation, and CGA is the anion. They're gel-type resins that you can purify things like saccharides or some small amino acids to large amino acids and small peptides. We have also the Chromalite MN. This is a hypercross-linked polystyrene adsorbent, very high surface area. Typically can be used as a replacement for activated carbon due to this kind of surface area availability. Chromalite PCG is a macroporous polydivinyl benzene adsorbent. When you talk about polydivinyl benzene, it means most of it is divinyl benzene, then styrene. And that is a requirement in the United States. If you're above 79.5% divinyl benzene, it could be used for food applications. So these kind of products could also be used in food fractionation, such as flavors and so on. We have the Chromalite AD. That's when you're starting to get very small for analytical purposes and preparative chromatography. It's based on the PCG chemistry, but on a much smaller scale. The Chromalite MDAE, which is kind of a new product, also from Purolite. This is mainly a polymethacrylate at this case scenario, because you want a little bit more hydrophilicity, because now you're dealing with a little bit larger peptide. You don't want non-specific binding occurring on the surface of the bead. So we would use a polymethacrylate, and then has a weak anion on it, a diethylamine, to do fine separations. For the Chromalite AD, if you don't want to buy them as bulk, we can offer them in pre-packed column, HPLC pre-packed column. And also, we can offer them as SPE reverse phase for people who want to work with the SPE product lines. So Chromalite particle size selection, again, we do have the 5 micron, mainly for the analytical HPLC application, 10 and 15 micron for preparative HPLC. And then we have various particle size that you could use for polishing, small molecule separations, whether it's small molecule or you're starting to get into the small protein size purification. Mainly based on hydrophobic interaction rather than ion exchange, a lot of these products. And then when you start getting into much larger scale, 75 and 150 micron, and you're talking about very large industrial chromatography application where you're talking about thousands of liters in a chromatography column. So looking at the product line and the particle size selections, you could see, product, I just remember doing this. You could see that we do have the cation resin, 10 micron, 50 micron, 200 micron, as well as the anion resins goes from 10 micron all the way to 200 micron as well. Reverse phase is what we call a non-functionalized adsorbent. So it mainly deals with hydrophobic interaction, how hydrophobic molecules could be attracted to the surface of the bead. And a lot of these products could be offered in a solid phase extraction, small little columns, if you want to do some SPE application. And like I mentioned before, the methacrylate, MDAE, which is a kind of a new product from Purolite for fine separation of small proteins and large peptides. So the porosity of these products also vary. For example, for the non-functionalized resins, I mentioned our mean particle size MN. It has a very high surface area. To have a high surface area, you mean you have to have a very small pore structure. So you can see that the MN is available as 30 angstrom pore diameter. So you can achieve above 1,500 meters square per dry gram surface area for a very large amount of removal. So most of the time it's used for color body removal when you're dealing with small molecules. But then we have 75 to 200 angstrom pore diameter as a PCG 600 and a 200, 300, PCG 900, and so on, and PCG 1200. So every single pore structure is offered for your separation in here. So it doesn't really matter what the molecular size of the product you're working with. We do have a product that will do the purification for you. So now for people that don't know which product might be the best one for them, because I'm dealing here with at least 6 different polystyrene divinyl benzene with different surface area, different pore structures, and so on. We do also offer a screening kit for companies that don't want to inform Purolite of any of what they're doing, and we can offer you the screening kit for your for your evaluation. But the more you can tell us about what you're trying to do, the more we can assist you. We do have the technical support that we can recommend certain resins. We can also customize some of these products to your application. And you can see that the effect of some of these pores on what they're excluding as molecular weight of the compounds. As I mentioned, we do have the SPE reverse phase. So these are small columns with 1 milliliter to 6 milliliter worth of resins that people can just do a very quick screening, or if they're doing in a discovery, typically that's what they work with. It's a one-time use, extraction, quick extraction, and so on. You could have any of our adsorbents packed in there, or you could have some of the ion exchange packed in there. You just let us know about what your interest in the SPE application is. So talking about the effect of Chromalite and the Purophase, because they're both combined together as SPE or bulk. So if it's sold bulk, we would sell it under the name of Chromalite. If it's sold in a SPE, would call it under the Purophase name. You could see some of the pore structure. It's not just important what the pore structure is. What is also important is how fine-tuned the pore distribution is. Because if somebody says to you, oh, I have a very high surface area, if your molecule cannot get in, you're not going to be able to remove it. So choosing the right pore structure or the right surface area will maximize the efficiency and yield of your purification. So you could see one of our products that's listed here is the PCG 1200C+. Very narrow pore distribution allows you to remove things at a very high capacity as long as it's that size. We did some work with for certain customers as well. This is one of the screening we've done for companies. They would send us sometimes their feed, or they would tell us, we're trying to separate this, give us your best options. So in this case scenario, they didn't give us the feed. They just told us that doing a paracetamol purification, they want to have the highest yield possible. So we did screening, and we found out that the PCG 600M would give them the highest yield, also as well as the highest capacity in running the process. But most of the time they do send us their feed, and because one of the important things is it's not just important that it has high capacity for paracetamol, it also have to have high affinity towards paracetamol versus other compounds or impurities in your solution. So if you are able to share your feed, we will be able to let you know the affinity to your molecule versus others as well. So in the Purophase, we did some SPE quick testing, and we tested paracetamol also yield, and you can see that we evaluate all the blue ones are the Purolite products, and all the green ones are the competitor products that were evaluated. So in this case scenario, you can see the PCG 600M giving you the highest yield between all of them. For vancomycin, it's another antibiotic, same exact thing. People came to us. They were dealing with a different product at that point, and you can see what product, which is HP20 at that point, which is manufactured by Mitsubishi Chemical. They approached us and they said, do you have something that can give us a little bit faster kinetics to load, get the things off, and also at a higher capacity, because they don't want to invest in a larger column. They just want to use the same column they purchased, and or they have it in their system. So we did a few testings for them, and we found out rather than having an HP20, which is a large bead, because they were using a too large of a bead, we got them to use a smaller size, 75 to 150 micron, which is our C, the PGC 600C, PGC 900C, give them a very quick available surface area for them to load and elute. And you can see what they were able to achieve, loading above 50 grams per liter on resins versus the HP20, higher yield and higher purity as well. So they ended up switching at that point. The price difference is not that much between them, between the large bead HP20 and the PCG 600C. So it wasn't that much of a cost difference for them to make the move. Also, one of the issues of making things separate better is you have to go smaller and smaller in particle size. So that's one of the things that Purolite did. Everything is successful on a large bead size, they try to make a much smaller size. In this case, you see the 15 micron that was created. That was actually made for insulin applications. You could see the data here, mentions the pressure stability. Going at 3 millimeter per minute flow rates, you could actually get a pressure of 200 bars and still is linear. So you don't have that compression that you would get from other type of polymeric matrices. So this is the data about the insulin purification. It's 6,700 Dalton molecular weight, and that was we did it at a 15 micron and a 35 micron to show that you could scale up this insulin purification. You see mainly two peaks here, which is the insulin, and the other one is the desamido insulin, which typically you can make by heating up the insulin, and it'll create that impurity. And that was compared against 3 of the major companies we compete against in this industry, which is Mitsubishi, and Amberchrome, which is now Dow. It used to be Roman Haas at one time, and YMC. And if you're not doing 15 micron and you want to go all the way down to analytical size, we can offer you our packed columns. Anything you do on a large scale, you could duplicate on a small scale, and anything you do on a small scale, you can duplicate on a large scale. It's Purolite is mainly focused on ion exchange resins. That's mainly what that company does. So you can imagine being the second largest ion exchange resin manufacturer in the world after Dow. It makes us, a right partner for a lot of our accounts. We do have a lot of production facilities. The two that are of interest to you are the ones that are making in the life science industries. So we have one that mainly does the development and R&D located in the UK. Typically, they can scale up to 5 kilos. And then in Romania, we can scale up even above 1,000 kilos if needed per batch. So, and 3 of the product lines are made there. The Chromalite resins, the ECR resins are made for enzyme immobilization. And then also we can immobilize enzyme on the resins at that facility as well. So like immobilized lipases and proteases are also available on a large scale. So we just built a new mini clean room, mainly to support customized Chromalite production. Some companies would like us to change certain physical or chemical properties on the resin, and we can actually do that. We can, functionalize any functional groups. We can dry it, pack it, and ship it. And if everything works well, then we would scale it up to the larger facility. Also in August of 2015, we actually invested quite a bit in the laboratories in Romania to support screening several resins and so on, and support our customers with their purification needs. So, you don't see that many chemists right now there because this picture we're taking as we were putting everything in the laboratory. But so we invested quite a bit to do this kind of screening. We do this kind of screening either free of charge if there is a immediate replacement requirement, but we also would charge if it's something that you want to do a feasibility study. So we would look at the option and let you know if that's feasible or not, and if it's scalable or not. And in our pilot scale manufacturing, we could make up to 10 kilos of the final product. Let's say you do like some of the customized resin that we've made for you in a lab scale. We can scale it up to 10 kilo. You could see that it is scalable. We could make it food grade production. So if it's a food customer, we can actually do that for them. And it's also vacuum drying, which is also important for the people that are used biocatalysis, that the enzyme is dried when it comes to them. So what Purolite can offer for you is one of the widest platform of synthetic polymers, methacrylates, styrene, divinyl benzene, but also the agarose that I mentioned at the beginning. Tomorrow we have a presentation also for Purolite done by Dr. Carolyn Tinsler, and she at 10:30, and she'll be presenting about our agarose for large molecular weight and antibody purification. Purolite also makes the widest range of particle size of resins from 5 micron all the way to 1200 microns. The larger bead are usually used for catalysis, large-scale catalysis application. And then we have, one of the best technologies in the market. We act quickly in customizing resins and supplying new products. In technical support, we have a team of 15 chemists in the UK that are there just to assist worldwide questions about a lot of our life science, resins. We do toll manufacturing if needed. We also have FDA-approved resins. And we follow regulatory, whether it's FDA, RESAP, and our plants are all halal and kosher certified. Like I mentioned, tomorrow, 10:30, our Presto agarose resins for large molecule purification, such as proteins, will be presented by Dr. Carolyn Tinsley, and it will be also in this room. Please show up, and if you have any questions, our stand is joint with Midas stand at CH11. Okay, thank you.